Welcome to the lecture series on vitamins and nutrition. Today we are going to look into one more category of fat soluble vitamin, that's vitamin E. So, if you look at vitamin E, it is a very interesting vitamin, and it might appear in the outset that this vitamin may be associated more with cosmetic related applications, because you find plenty of vitamin E related oils and preparations available. It is one of the most popular vitamin, not as a dietary supplement, but mostly as a kind of I would say formulation aid or as a kind of cosmetic aid. But if you closely look into the function of vitamin E, its contribution is really essential. It is one category of vitamin where, apart from its nutritional utility, this vitamin has a wide variety of application. One more vitamin which comes under this category is the other water soluble vitamin, vitamin C. We will see about vitamin C ascorbic acid later in the sessions, and today our discussion with vitamin E and vitamin C, when we always talk about antioxidant vitamins, we normally synonymously use vitamin E and vitamin C, because their role in antioxidant function is well appreciated in variety of preparations. So, our focus today would be on vitamin E. We are going to look into the chemical aspects of vitamin E, starting from its historic origin and what are the structural features of vitamin E, its biochemical role, then how this vitamin E is going to contribute physiologically in variety of functions, and many more associated with vitamin E. And as we have been looking in the earlier sessions, vitamin E generally is one of the most essential vitamins in terms of maintenance of healthy physiology. So, in contribution with other fat soluble vitamins, its role is very important. So, if you are going to look at vitamin E, immediately you think of vitamin E as a fat soluble vitamin, and it has a hydrophobic oily texture, and being a fat soluble vitamin, it is actually associated with sterility and fetal death, because the early experiments of vitamin D were done in rats. So, when rat, one of the most common mammalian model to study about human system, most of the experiments and toxicity studies, we subject the chemical in rat models and in mice model. Sometimes most of the functions what we try to derive from fat soluble vitamins in mice or rat model are mostly found to be almost 90% good, but there are some exceptions to the rule. Say for example, for vitamin E, in rats and in mice, deficiency of vitamin E caused sterility, and its role in fetal development was extremely essential. In fact, if you deprive mammalian system of lower organisms, in this case the rat and the mice model, there would be fetal death and there would be spontaneous abortions, and that is how people drew their attention to the study of vitamin E. Even now, even in human beings, its role in sterility is essential. Although you cannot completely extrapolate the kind of abortive features what is seen in the mice or rat model to be true in human systems, but nevertheless a continuous deprivation would obviously lead to all kinds of deficiency conditions, and that can eventually lead to fetal death. So, if you are going to look at the technical term of vitamin E, it is a very interesting term. So, it is called tocopherol. So, toco means in Greek childbirth. So, as I have been telling you, since this particular vitamin's use was studied way back since 1922, people were conducting lot of animal experiments starting from Funk and many people, and this particular scientist needs to be mentioned. His name is Paul Carrer. So, Paul Carrer was a important scientist in terms of his contribution in elucidating vitamin structures. In fact, he is the one who elucidated the structure of provitamin A in detail and all the carotenoid related structures. His contribution to flavonoid chemistry was tremendous, and in addition to that, he is the one who is given the credit of elucidating the structure of vitamin E. Not only that, he also contributed in elucidating the structure of vitamin C, ascorbic acid. So, Paul Carer's work led to the functional role of vitamin E in detail. Before that, people could not really associate the functions or the biochemical role that it played, but they were normally attributing the role mostly to B category vitamins. So, after Paul Carer's elucidation of vitamin E structures, it became much clear for the scientist to really understand the role of vitamin E and associate the role of vitamin E exactly with the biochemical function that it is playing in our body. So, if you look at it, Phero means to bear. There was fetal death and fetal abnormalities and all sorts of sterility related conditions happening in animal model. Vitamin E was given the name tocopherol. Being a OH group is there, it is basically a alcoholic derivative. So, you consider vitamin E to be a anti-infertility vitamin. So, it was very interesting to note whenever vitamin E was supplemented to those animal models, the mice and the rat model, when they were deficit of vitamin E, when they showed symptoms of sterility, the conditions could be reversed and the conditions could be made more fertile. In fact, with sufficient supply of vitamin E, the sterility status of these rodent models were reversed and they would be able to reproduce well, and their role in terms of healthy skin texture was also tremendous. So, now we are going to look into different functions of vitamin E. As I have told you, vitamin E is given as a name to a category of basic structure, what we consider it to be the tocopherol nucleus. Because if you look at the kind of tocopherol nucleus, you do have subcategory of tocopherols, and we give this Greek alphabets to differentiate each of the structure. So, what we call it like alpha, beta, gamma, delta, and it goes to a point you have around 8 category of tocopherols. So, if you look at the structure of vitamin E, so we say tocopherol, an alcohol molecule, and you have tocotrienols, you have more polyhydroxy functional group present in the vitamin E. Now, all tocopherols have a primary ring, what we call it like the Croman ring, and all the subderivatives tells you the subtle differences you find in the side chain. So, what are these side chains made of? If you recollect, in some of the earlier sessions I told you all fat soluble vitamins should have a long chain hydrophobic tail like appearance as a side chain, most of them. Vitamin A we saw, and in case with vitamin D we saw the entire hydrophobic phenanthrene ring was there, and in vitamin E we find the same side chain. In this case it is a isoprenoid unit. So, if you look at the isoprenoid unit, you consider this to be the structure. So, if you look at the structure, you have the tocol nucleus where you have 2 rings, one is the benzene ring with alternate conjugate bonds, and then you have a heterocyclic ring with oxygen over there as a heteroatom, and this ring combination is basically called the tocol. So, this tocol combination, you have different names depending on the substitutions in the side chains of the tocol nucleus. R1, R2, R3 shows that you could have different varieties of methyl group. If all these structures are methyl group, you consider them to be alpha tocopherol. So, all these are methyl group, and the side chain as you see over there, you have a isoprenoid unit, and the isoprenoid unit gives you that essentially the lengthy tail like appearance in the vitamin E structure. So, if you look at what are those different categories, so among the 8 categories you have 4 more prominent category of tocopherols, and you have the methyl group, what we call it like trimethyl group, as you have seen over here, the trimethyl group R1, R2, R3, and in case with beta tocopherol you have a dimethyl group in the 5th position and in the 8th position, and then you have a gamma tocopherol, again it is a dimethyl derivative with hydroxy group with a methyl group in the 7th position and 8th position, and in case with delta tocopherol it is a monomethylated derivative only in the 8th position. So, you see here with more methyl groups in the structure of tocol nucleus will contribute in increase in the activity of vitamin E, although essentially all these 8 category of vitamins definitely have a biochemical role to play and their function is almost synonymous. Now, we are going to look into why you have these differences. So, in other words, what we are trying to look at, why these vitamin derivatives basically show differences. So, here you have pink star mentioned over here, tells you the chiral center. So, what is a chiral center? You would have studied in organic chemistry. When a carbon atom is attached to 4 different groups, the carbon has chiral property. It means it has the probability of being a stereo center. So, the moment you consider a stereo center, the 3-dimensional structure of the molecule can vary, and this would definitely contribute in the way they interact with endogenous receptors or exogenous receptors present in a body, in the cells especially. So, this chiral groups, some of the chiral groups can actually give rise to a 3-dimensional structure which may not be effective. So, we have to look at the biologically active form. So, all chiral structures need not be biologically active, but the one which we have mentioned over here tells you about different substitutions that you can look for in the chiral center. So, you see over there the alpha, beta, gamma, delta. Apart from the tocol nucleus substitution, the chiral center, what we have mentioned in this slide, also gives you the way by which the side chain can also participate in the way they interact with the receptors. Now, we are going to look at different versions. You see here alpha tocopherol, gamma tocopherol with different substitutions over there, and you find the isoprenoid unit with multiple methyl group substitutions, and here you have in principle the alpha, beta, gamma, delta category of tocopherols. Now, if you look at this particular vitamin, it has oily texture. In fact, the consistency is, I would say, very viscous, and hence even vitamin E as such can be used as a topical emollient in case of burn injuries and skin toning related functions. So, it is one of the essential ingredients in many kinds of skin or dermatological preparations where you need to create an antioxidant barrier, and if you want to have a soothing function and you want to prevent dry skin. So, you see over there it is a kind of a yellowish oil, it is quite heat stable because being a hydrophobic entities and hydrophobic components present in the structure, it is quite stable, and but it is little labile in the presence of alkali, and it is extremely labile for oxidation. In fact, that is the reason why you use vitamin E as an antioxidant, because this would preferentially get oxidized and prevent the system or prevent the compound for which you are adding vitamin E as an antioxidant. So, it gets preferentially oxidized. So, it is like very labile for oxidation. So, you see there low molecular weight, that makes vitamin E a very good choice for most of the topical dermatological applications. And as I have told you, they have optical activity because of the chiral center, and you see over there they absorb UV light. So it could be incorporated as a pharmaceutical aid in many of preparations where you are going to use UV related radiations for therapeutic interventions. So you see over there, you can make salts with the hydroxy group present in the chromone ring, and here is a quick look of all the sources by which you can get vitamin E. So I am going to elaborate more on these sources in the coming slides. So you see over there, the first three choices happens to be oil: cottonseed oil, sunflower oil, corn oil. Always consider there is this natural ingredient of vitamin E already present in these oils, but the ones which you buy commercially, most of the oils which are used for cooking purposes are probably fortified with additional doses of vitamin E, vitamin A and vitamin D, as I have been telling you earlier. In addition to vitamin A and vitamin D, vitamin E is also added to enhance the properties of the oil. So you might wonder why vitamin E have to be there as part of these vegetable oils. Most of these vegetable oils are unsaturated oils. In other words, they contain unsaturated fatty acids. So unsaturated fatty acids are more susceptible for air oxidation. So if you could have vitamin E in these oils, it not only improves the overall quality of the oil to have an antioxidant function, depending on the property for which, or depending on the use for which the oil is going to be utilized. In addition to that, it would also act like a natural preservative, preventing rancidity of oil. So we are going to look at rancidity of oil in the coming slides. I will keep going. So you see over there the three sources: cottonseed oil, sunflower oil and corn oil. So when you look at these oils, it means in addition to their triglyceride content, long chain fatty acids and other vitamins present in them, vitamin E is also there. You also have soya bean, a good source of vitamin E. So in this case, I would like to mention soya bean being an excellent source of vitamin E. Not only vitamin E, there are many steroidal components, or phytosterols as we call it, is present in soya bean milk. Then you have cabbage, then you have wheat germ oil. It's interesting to note the fish liver oil does not have vitamin E, but it's rich in vitamin A and vitamin D. So the trend is, when we do fortification or when we recommend dietary counseling, we ensure that we need to take wheat germ oil as an additional component to fish liver oil. So that way you would give a complete supplementation of vitamin A, D and E. Here is a quick picture. So I am going to run 2 slides and some of the pictures would be redundant, and I purposely did it to ensure that this would be the richest source for the oil. See, for example, wheat germ oil, then olive oil, then you have sunflower seeds. Always remember, the source of oil would be always seeds. Seeds are also the source of proteins. It's true, many of these seeds have to carry energy for the new life that they have. So when a seed is dropped for growth, it should provide energy, packed source of energy. So the best way to provide a packed source of energy would be to store the energy in the form of fat, fatty acid or oily substances. That's one of the reason most of the seeds happens to be an excellent source for oil. Not only the seed, because the kernel in the seed is the actual source of these nutritive substances. So human beings, we actually extract these nutritive sources, but if you look into the natural origin of why seeds have this extra source of energy, is because they should provide for the new life as the seed would germinate. So you see over there, most of them like peanuts, almonds, then even you have dates and dried tomatoes, could be a good source. So you see over there, you find something redundant, you find coconut olive oil redundant and sunflower oil redundant, you have almonds and peanuts redundant. So this redundant pictures tells you that they form one of the excellent sources of vitamin E. Now in addition to that, you do have broccoli, kale and spinach, the green vegetables being a rich source of vitamin E. And you also have avocado is one of the richest sources of natural saturated and unsaturated fatty acids. And always remember, vitamin E, vitamin A, vitamin D are all lipid soluble. So it's natural to look for any lipid soluble base in which these vitamins would naturally occur. So in that way, avocado and all these nuts being a good source, because they are good source for fat, and it's much easy for you to incorporate vitamin E and hydrophobic related structures along with the lipid soluble base. So now we are going to look at the recommended daily allowance. So I have been telling you, like, what is the average intake of vitamin E, or rather to say this way, if you are going to look at what would be the minimum requirement of vitamin D on our everyday diet, so that we will not show deficiency diseases. In other words, if they have a particular role to play, biochemical role to play, a physiological function to do, how much minimum or what is the exact requirement? See, this exact requirement is basically the recommended requirement based on average values. It doesn't even reflect the ethnic values, but overall, if we go beyond this value, either above or below, we are surely going to end up with some trouble. So there would be individual need to need basis of requirement, more or less, but this value would be a good guide for us to decide how much amount of vitamin should be made available for the person when they are under deficiency conditions or therapy. So you see, for male it will be like 100 mg per day. In the international units it's like 15 per day, and in case with female you have 8 and it's around 12. But if you look over here, so during pregnancy and lactation the requirements of vitamin E increases, because the body would now undergo changes and it would be able to make this particular vitamin available. The bioavailability for delivery would also be high in the presence of nutritive fatty substances present in milk, and all the reproductive structures require it in the early stages of development. So there is extra demand for the growing fetus, so the daily requirement should be like increased. Now you consider over here you have polyunsaturated fatty acid ratio of tocopherol. So you see like 0.6 mg alpha tocopherol per gram of PUFA, polyunsaturated fatty acid. So wherever you have this polyunsaturated fatty acids, you are likely to have vitamin E, because vitamin E would be closely associated with fat soluble bases. Okay, so polyunsaturated fatty acids being an excellent source for dispersion of vitamin E. Now here you have one more list, and as I have told you earlier, we have little more extended alpha tocopherol products. You see over there most of them like palm oil, rapeseed oil, olive oil, all the nuts being an excellent source of vitamin E. Then from the animal source, if you look at it, apart from sunflower and soya bean, here you have tallow, lard, the herring oil, the cheese, the butter, the margarine, and all these are also good sources. So you see over there corn germ oil and soya bean oil happens to be one of the excellent sources for vitamin E. And here you have an extended list. You have different I would say the portions of the animal organ meat rich in vitamin E. So you see over there the liver, the veal, the herring, the mackerel. So all these have very good source for contribution of vitamin E. Here you have again a list of all the vegetables with good source of vitamin E. So, when we say vitamin E, if you look at it, we are looking at alpha tocopherol, because as I have told you earlier, there are around 8 derivatives. So, we make all the calculations in terms of alpha tocopherol. Yeah, you might have other derivatives as well, but incorporating in terms of alpha tocopherol really helps. Now, we are going to look at the metabolism of vitamin E. It is quite interesting to look at vitamin E metabolism. If you look at all fat soluble vitamins, you do have always this limitation of bioavailability by nature of its lipophilic property. So, you need to really emulsify these vitamins before you could ensure they could cross the barrier, the gastrointestinal barrier, and reach systemic circulation. So, bile salts would be able to do that. So, dietary vitamin E. So, whatever I have told you earlier, all the food substances that we have seen, these food substances are now emulsified. The fat material is emulsified. So, being a lipid soluble vitamin, so presence of bile salts would definitely encourage good absorption of vitamin E. So, here if you look at it, the absorption is basically by diffusion in small intestine along with other lipids as micelles. So, we say the moment you see this word micelles, you should remember it is talking about emulsification process. So, vitamin E along with other fats would be emulsified, and this emulsified version finds its way in our systemic circulation. Now, among different kinds of, I would say, lipids that our body would synthesize in transporting these lipids to different parts of the body, generally there is a trend. They consider lipid or fat or triglycerides to be associated with cholesterol related diseases and coronary artery diseases and all kinds of vascular related diseases. It is true, but we also need all these fats essentially for insulation, essentially as a base for synthesizing variety of steroidal hormones. So, in that case, you need to look carefully how we are going to transport these lipids into systemic circulation. So, we are going to make a quick summary, and what we have seen till now is basically the chemical structure of vitamin E and their availability from different sources, and we have seen extensively how the chemical function of vitamin E was elucidated and what role it plays, and we have also seen what all the good sources of this vitamin E both from plant sources and from animal sources, and now we have stepped into the aspect of absorption. So, in the coming session we are going to deal elaborately on how absorption happens and what would be the metabolic role of vitamin E. Thank you for your attention.